Hello everyone, welcome to the Science Stuff. Today we're going to be looking at how to conduct a gram stain and also be talking about the differences between gram-positive and gram-negative bacteria. So what are we waiting for? Let's get right into it. A fun fact about gram staining is that it was discovered by a Danish bacteriologist, Hans Christian Gram. To start our gram stain, we're gonna need bacteria from either a plate or a broth that is heat fixed to a slide. We're gonna take the slide and add in our primary stain, which is an initial foundational dye that is used to color all microorganisms in the sample. We're gonna use crystal violet for the gram staining and add it to the bacterial sample and completely cover it for 60 seconds. After 60 seconds, we're going to rinse it over with water and then add in our mordant, which helps bind this primary stain more effectively with the bacterial cell. The mordant we're going to use is grams iodine. We're going to add it for 60 seconds and then rinse it with water as well. The next step is adding the ethanol, which acts as a decolorizer, causing the crystal violet iodine complex to dissolve away and rinse out of the cell wall. We're going to add this for about 5 to 10 seconds and then also rinse the ethanol out with water. For the last step, we're going to add in our secondary stain, safranin, which acts as a counter stain in the final step of gram staining, coloring the previously decolorized gram-negative bacteria pink or red. It's important to note that gram stains are still widely used in hospitals as a fast, essential, and low-cost diagnostic tool to identify bacterial infections, guide initial antibiotic selection, and determine if bacteria are gram-positive or gram-negative. So we've talked about how to conduct a gram stain. But what really are the major differences between gram-positive and gram-negative bacteria? One of the major differences is the color of the bacteria after the gram staining. Gram-positive bacteria appears blue or purple as seen here, while gram-negative bacteria appear pinkish red. Now we can relate this back to our gram staining, as the blue or purple comes from the primary stain that we used, crystal violet, while the pinkish red comes from the secondary stain that we used, safranin. Now why does this happen? The large variation in the color after gram staining is mainly due to the differences in the thickness of their cell walls. Gram-positive bacteria have a thick cell wall consisting of many layers of peptidoglycan, while gram-negative bacteria have a thin cell wall with only few layers of peptidoglycan. Gram-positive cell walls are about 20 to 80 nanometers thick, while gram-negative cell walls are less than 10 nanometers. The crystal violet, which was the primary stain we used, binded well with the thick peptidoglycan layer and was not washed out with the ethanol while we tried to decolorize it. The gram-negative bacteria, however, due to it having very few layers of peptidoglycan, was easily decolorized and stained with the secondary stain, safranin instead, giving it its pinkish red color. Another difference that is observed is that gram-positive bacteria have these anionic glycopolymers called teichoic acids in their cell walls that gram-negative bacteria don't have. There are also noticeable differences in the toxins that are secreted in gram-positive and gram-negative bacteria. Gram-positive bacteria secrete exotoxins primarily, and although these toxins are capable of causing injury to host cells and resulting in disease, they require a significantly lower dose than what is required by endotoxins. Endotoxins are heat-stable lipopolysaccharide components, and these lipopolysaccharides are not found in gram-positive bacteria. They are only found in gram-negative bacteria. They are released mainly upon cell death or lysis and cause severe immune responses in humans, including fever, inflammation, and potential septic shock. The difference in the structure of these bacteria also makes them respond differently to antibiotics. Now, gram-positive bacteria don't have the presence of an outer membrane. This is a primary feature of gram-negative bacteria. Now, this outer membrane, specially containing lipopolysaccharides, acts as a crucial barrier that excludes many common antibiotics from penetrating and reaching their targets. This means that gram-positive bacteria have a higher susceptibility to antibiotics and are less resistant, while gram-negative bacteria are more tolerant and can resist antibiotics. Some important examples of these bacteria can include Staphylococcus aureus, which is a gram-positive bacteria that is commonly used in research. There's also Streptococcus pneumoniae, Enterococcus faecalis, and Bacillus subtilis. Some examples of gram-negative bacteria is the model bacterial organism E. coli. There's also Salmonella enterica, which causes salmonella, Klebsiella pneumoniae, which causes pneumonia, along with Streptococcus pneumoniae. Both of them can cause pneumonia. And then there's also Shigella dysentery, which can cause dysentery. Thank you for watching this video. I hope you now have a better understanding on gram staining and the differences between gram-positive and gram-negative bacteria. Please like, share, and subscribe, and comment down below on any questions or concerns you may have. And remember, you're watching The Science Stuff.